It's me great pleasure to welcome Dr. Gary Samuelson to the webinar. Dr. Gary Samuelson, for those that you don't know, has a medical and atomic physics degree from the University of Utah and has dedicated his career and knowledge to the advancement of technologies that address the major problems of health today. His focus is on the science of redox signaling that is now developing around the world. Dr. Samuelson is the one and only scientist who was able to stabilize these molecules outside the body for our human consumption. Dr. Samuelson, are you ready?
Thank you very much, good evening to all of you. It's a great pleasure to be able to be with you this evening and to talk a little bit about some of my favorite topics in science. It was a wonderful thing that the ASEA company has called the ASEA supplement now, or the ASEA REDOX supplement, a cell signaling supplement. And that is because the cell signaling processes that we have inside of our body are some of the most important processes that we are able to sustain health and the functionality of our whole body. And if you think about it, you know, we ask ourselves, well, how does our body work? How in the world does this great machine, this incredible and marvelous machine of ours actually function? We've heard certain things like the DNA and the genetics, and we understand a little bit about that. We understand also that the signaling systems that we have in our body, about the brain, the digestive system, and other such things, and how they work together, the circulatory system, and how they work together in order to be able to create in our machine something that will sustain our lives. And then, of course, as an atomic physicist, I have a little bit more interesting perspective on this. We go down past the size of a cell, past the point where we're looking at these cells that are constituents of our body, to the point where we look at what's happening inside the cells, what is happening on the cellular level, to have all of these things function together perfectly. And as we mentioned before, we have tens of trillions of cells in our body, tens of trillions of cells. That means that just in the little point of my finger right here, I have more than 1 billion cells. And all of those cells need to be able to coordinate their activities and work together in order to be able to form a living organism. And imagine the amount of cooperation that we must have between ourselves. How about just the type of coordination that is needed for one heartbeat? In our heart, we have billions of cells, and all of these cells are connected together. We have two places in our heart, one on the very top where the signal comes in and starts the contraction of the aorta, and then another signal which goes and starts the contraction of the ventricles. And the coordination of those signaling mechanisms needs to be perfect in order to be able to force the blood from our lungs and the rest of our body through the rest of our body. Imagine the signaling process that have to take place among billions of cells in order for such coordination to take place. And it all has to do then with cell signaling. Also imagine in our brains, we have billions again of neurons. And each neuron is connected with the other neurons in our brains through these little synapses. And the signaling processes that take place between the cells in our brains actually give us the ability to reason, our cognitive ability, the ability to be able to have all the functionality of our brains, our memories, our past experiences, our emotions, the ability to recognize patterns and to associate these patterns with things that may be dangerous or things that may be familiar to us. And all of these things are happening inside of our brains by cellular communication. If I were to raise my hand, the signaling inside my brain, which on an atomic level could be on the other side of the universe almost, from the muscles inside of my arm. The signaling processes on the atoms in my brain are actually causing these signals to go through these tissues to target specific muscles in my arm, and therefore I have control over my body. So that's also a signaling system. And then all of these signaling systems throughout the whole body need to be coordinated through our cells. Each cell, then, as we have said, allows signals to come in. Some are hormones that are coming into our cells on a daily basis. Some are electrical signals. I like to think of each cell as being somewhat like a house in a community, and the hormones that come in are like the mailboxes. They're the mail. And the streets of the house are like the blood vessels that carry the supplies and everything that's needed between cells. And then the electrical signaling processes also happen between neuronal material, neurons, or conductive material that's between all of our cells. And so what we find is the same as our brain cells are all connected together and signaling, every cell in our body is also connected together and signaling. And the processes that keep these signals then working and viable are extremely important in our cells to maintain the integrity of our whole body. In fact, we almost might say that we are the product of signaling cells. That's what makes us alive. That's what gives us life. If there was no signaling between our cells, we would be, in effect, dead or unable to do things. So these signaling processes are very extremely important. I wanted to talk a little bit now about genes, since I am going to go into in a little bit of depth the study that took place. It's taken over 2 years in the Tourette laboratories in Salt Lake City, Utah, about how these redox signaling molecules then affect our genes, and which genes then are affected by these redox signaling molecules, and what the significance of that could be to the signaling processes that happen in our body. And before I go into the genetic results or the genetic things, I wanted to explain to you a little bit about why and how our genes work. You might think that our genes are the things that give us our eye color, that give us our skin tone, our facial structure, our bone structure, the things that give us our physical characteristics. That is very true, but that is only a very small part of what the genes do. Inside our bodies, the genes every second, every millisecond, millions of times a second, are being expressed. What do I mean by an expression of a gene? Think of our little cells then as little tiny micro machines, as tiny little computers. Or you might want to think of them as cell phones, because we know we have smartphones, and we can hold them in our hands. And these smartphones that we have applications. So if we wanted to dial a number, for example, I push that little button that has a phone on on the screen of my smartphone, and it pulls up a dialer and then allows me to make a phone call. Every single cell has these applications written inside our genes. There are some 30,000 genes in every cell, give or take. And these 30,000 genes then are activated in order to carry on the processes of life inside our cells. by the signaling messengers that happen to come by. And then that's a really good question. Now, what actually activates our genes? You might want to think of the thing that activates our genes as a huge our genes as a huge panel of buttons, let's say, and each button then can activate a certain gene and activate a certain protein inside of our body, or the production of a certain protein. And so the question that I asked is, who is pressing these genetic buttons that keeps our genes active? And the answer is actually very interesting. Almost everything presses these genetic buttons. For example, if we have a happy thought, our happy thoughts can actually express genes which make hormones like serotonin inside of our brains, and the serotonin and such is a hormone that gives us this feeling of elation, of feeling good. And just a happy thought, for example, expresses these genes and expresses the hormones inside of our system. The genes are necessary; those genetic buttons are necessary to produce the hormones that are used to control our moods. So moods can affect genes. What we drink can affect our genes. If I drink some orange juice, for example, and the components of the orange juice, the acidity and such of the orange juice goes inside my stomach and is digested, well, that affects my genes too. So the question is, almost everything affects my genes. The question is then how does the body know which of these genetic buttons to push? Stem cell technology is also growing very big. Stem cells are cells that do not have an operator that presses specific genes to make the cell operate as a heart cell or a brain cell. They can have a specific function in our body because certain genes are pushed in the heart cells that transform them into heart cells. And certain genes then are pushed also that transform them into a brain cell or whatever cell they happen to be around, these messages that are being passed back and forth. So the expression or the activation of these applications inside of our cells can be affected by many different types of things in our environment, from the thoughts that we're thinking to the physical activity that we have. These genes are turning off and on all the time, and regulating a great deal of activity. I mean, that's very interesting. It has given birth to a whole study of genetics called epigenetics. Epigenetics means things beyond the genes, things that push the buttons that control the genes, the messengers that control the genes. And we ask ourselves the questions: what are they? What's their significance to our health? And how then do we actually map out exactly which genes are pushed? And so, as we have been mapping out the human genome, we've also been developing technology to be able to see through our blood sampling, through blood samples, which genes have been pushed and expressed inside of our cells. The ASEA study that was done then over this 2-year period of time started out as an idea. The idea was, in order to understand how ASEA works inside our body, why don't we look at all of the genes? And we now have little gene chips that are able to look at the expressions of tens of thousands of genes at a time from just one blood sample. And the expression of these genes. Let's look at them and see what is happening in these genes. And thank heavens, over the period of time, and as genetics has evolved, we're starting to understand which genes then press which buttons. or I mean, sorry, which buttons are press which genes, and how these genes then interact and send messages to the whole system. And this is very important information that has been built up. You can imagine the complexity, because there are 30,000 buttons out there, and the complexity of which are pressed, which buttons are not. So the Affymetrix people have developed one of these little chips that can monitor which genes have been pressed. Now we have to understand that the expression of genes in each person is a little bit different. You also have to understand that when people are going about their daily lives, eating, drinking, having a good day, having bad days, their genetic expressions are varying. So it becomes fairly complex in order to understand, well, which genes have been expressed differentially, or when somebody is taking ASEA, and which have not. And we have to have what they call statistic significance. So if we were to do an Affymetrix panel, of which we did, we would have to run a great deal of statistics to see which of those genes then were preferentially expressed more than other genes inside of our body, due to the random variations that happen all day long in all of us. So imagine the complexity of such type of analysis. And I'm very lucky often to be able to tell a difference. Now, ASEA did this with the Affymetrix panel. They took groups of 60 people, and they broke them into 3 types of groups. One was the control group. The control group just drank over the 8-week period of time. They just drank salt water. The group, the ASEA group, took the study group took the ASEA for this 8-week period of time, and then there was another group which was also a bit there, which took nothing. They didn't change into anything at all. In order to be able to show statistical significance in a change in the genes, the people in the ASEA group would have to consistently show that certain genes were expressed more often than the genes in the other 2 groups, the people in the other 2 groups. And this was done, and the significance was established very high in some cases, and in some genes that certain genes, when you drink ASEA, do become expressed much more frequently than the genes of the people that have not taken ASEA. And this is a very important result. And the question then that we can ask, well, what genes are they, and what does it mean? What does it mean to us? Well, the interesting part of this is that they were able to establish 5 genes that have statistical significance. This is actually a blessing. It's actually wonderful, because those 5 genes now have specific roles inside of our bodies. If we were to identify more than 5 genes, let's say 20 or 50 or 60, as we did in the metabolomic study a little while ago, it would be more difficult to specify exactly what ASEA is doing genetically inside the body. But in this case, it was very clear. What ASEA is doing is expressing the genes that help to facilitate communication inside of our cells. These are the most important genes that we have. Connected computers equal to the system of connected cells in our body that have specific things that they need to do in order to keep the body healthy and alive, such as those cells which detect damage in our body, or let's say the immune cells that help defend our body. These cells need to have the communication systems up and running. The blood vessels that regulate the blood pressure inside of our body, and the digestive system that needs to communicate. When these communication channels are down, our body can suffer significant problems. In fact, what we've said all along is the redox signaling molecules inside of our cells help the cells to detect the damage, repair the damage, and replace the damage. And they send the signals necessary to the cells to do so, the surrounding cells. And we've known that they're effective in the immune system, that they're effective in the vascular system, and in the hormonal system also. We've known that through our observations from separate studies before. We also know that they help protect our body. Remember the LDL cholesterol, the oxidized LDL cholesterol. It lowers the oxidation level of the cholesterol, which it helps our cardiovascular system. It also lowers oxidation levels in our body. It lowers the oxidative stress levels. We've known that all along. We haven't understood why this does that. And now the specific genes that were identified in the Tourette study are giving us a picture and a window to not only verify that these things are scientifically valid, but also to give us an understanding of why the ASEA product is doing what it does, that we have observed inside of our body. So there are 5 genes, as I mentioned, that significantly shifted in the ASEA group. This is very important. These are the very genes that help us to do 5 areas of functionality. And this has been mentioned before, but I'll mention it to you again. In the immune system, in the innate immune system, when we have damage, it builds up oxidation in our cells. That oxidation sets off certain genes that identify and help to help the cells to recognize or detect that the cell has been invaded. Now, this innate immune system is very important because it's the first response that the body has in order to be able to combat the bacteria that is in our body. The genes that are pressed, by the way, by this innate immune system when a cell are very important. They're the communication genes that help the rest of the immune system work. Are those affected by one of the genes here, the ERG1 gene that helps the system? It connects the systems together and helps to do the response that we see, been identified in the ASEA product. So we can say that ASEA helps the innate immune system to detect pathogens, to detect problems that may have happened when our cells have been damaged or attacked, and then also helps to facilitate the response of the immune system as this goes forward. There are other signaling that happen inside of our vascular system. I mean, this is absolutely very fascinating. When we have inside of our blood vessels problems like an increase in blood pressure from our heart or such, the blood vessels actually detect the increase of blood pressure and then dilate. They shrink and have to have good elasticity in order to stretch and to adjust to the blood pressure that our body has. So as we start to exercise, for example, our blood pressure goes up, remains then have to be able to adjust themselves throughout our whole body. This is also a redox signaling process. It's one of the first ones that actually we identified, and it's very, very interesting that identified in the study are those that help to maintain the elasticity and the response of the inner lining of the blood vessels in order to be able to keep our vascular tone and vascular elasticity during different changes of blood pressure and changes in oxygen levels, etc. All of those are regulated through also these stress levels, these reactive oxygen species, the same types of chemicals that are inside ASEA, that are produced by the linings of the blood vessels. Our hormones also in our brain also respond due to the oxidative stress levels inside of our cells. Oxidative stress levels excite the genes. The genes then are able to make the system adjust in order to be able to do what it should. The adjustments often mean that the cells have to produce hormones. Let me give an example. For example, if we have elevated sugar inside of our body, there are certain pancreatic cells that become stressed. They're called beta cells. They become stressed because they are burning, their mitochondria are burning much, much brighter because of the high sugar levels inside of our blood than normal. And because of this oxidative stress level, they push the buttons, the genetic buttons, to produce insulin. They express the insulin hormone. The insulin hormone then goes throughout our body and is received by other cells, and it instructs other cells to burn the sugars or to store the sugars away as fats, to convert and store them as fat. So you see, that's also an oxidative process that is control of the redox signaling process. And again, the genes that allow the body to respond to oxidative stress and hormonal production are also those which are affected by the ASEA product, by the redox signaling molecules in ASEA. Inflammatory pathways also. The oxidative stress production itself creates inflammatory cytokines and such that spread out and inflame the tissues. ASEA actually reduces inflammation in these areas where the tissues have been inflamed. The reduction of inflammation is shown by the reduction of the expression of the gene. The gene expression goes down and does not express as much of the inflammatory response as people who don't take ASEA. Helps to reduce inflammation. Now we have proof that, or at least we have an idea, or we understand that it's possible that ASEA can do this, can reduce inflammation inside of our tissues that have been stressed. Now, the new thing that we started to realize through this study, the new thing that for me is kind of exciting because it's something that I actually did not anticipate. All of the other things were standard redox signaling pathways that we knew had been excited by ASEA, and now we have genetic proof that shows that they indeed are on a genetic level being expressed. There is one that I didn't know of, and that was the production of digestive enzymes and the signaling enzymes that are in the digestive system. And we've seen the effects of this. You know, people take ASEA for their stomach problems all the time, or when they have distress in the stomach, whatever. I do, at least, and get get quite a bit of relief, and I never knew why. And we also found that it helps to both acidify and put more alkalinity in certain types of tissues in the body, and we've seen that in other studies that scientists have done. I never known quite why, and now we understand a little bit more that the one of the specific pathways of ASEA is to help to regulate the correct digestive enzymes in the digestive system. So isn't that interesting, and isn't it wonderful that after going through these 2 years of studies, we've identified exactly those genes that we've seen both clinically in our past studies and also observationally in thousands and hundreds of thousands of people now that are taking ASEA worldwide, are affecting the exact same systems that help to reduce inflammation, to help the immune system to become much more effective, to help increase the integrity and elasticity of our vascular system, to help the inflammatory pathways, as we mentioned before, and the hormonal production that is also expressed when we have certain stresses inside of our body to become normalized or more effective, as well as the digestive enzymes. All of these things now make sense. And so thank heavens, for the first time now, I can actually kind of breathe easy, and that we have verifiable science, that we can now hang our hat on something that says these genes are the ones that are expressed. These are the communication genes that are inside our body. These are the genes that keep the routers running, keep the Wi-Fi up, that keep the communication pathways running, especially in the immune system and in our hormonal systems. These are the genes that are probably the most important genes that we have in our body, because they regulate the communication pathways. They regulate the signaling pathways. So now we can say without equivocation that ASEA REDOX is a signaling supplement, proven scientifically to activate the genes that cause these signaling pathways in our body to be activated, as we've expressed them before. And so I've seen my little brainchild, the redox signaling, grow up, and we've seen it verified. We've seen it bless the lives of thousands of people across the world, and it is a beautiful sight to see. And I'm grateful to each one of you that are here tonight with us. And I'm sorry that some of us were not able to get on. I'm grateful to you too, also, for understanding the importance of this new technology and putting your efforts into getting it out to the people that can benefit thereby. Thank you very much. I hope you're still with me, Maria.
Certainly, I am Dr. Samuelson. Thank you so very much. What a wonderful lot of information you've given us. All brand new research, and I think, as you said, we can now hold our head high and just say we've got something amazing to share with everybody. And of course, we'll owe it to yourself. So thank you so very much. Now, we would like to open the lines to perhaps questioning. There are about 300 people on the call. So if we can just do this fairly orderly. If there are questions that you think require a little bit more in depth, perhaps you can put them in the chat box, and then perhaps maybe if we don't get to you, Dr. Samuelson can then get back to you through your upline at some point. So if we can just do it in an orderly manner, Dr. Samuelson is the driver, so he will do his very best to answer all the questions. And just as a reminder, can we please make sure that the questions are compliant with the ASEA policies and regulations, and also just the note that ASEA is not a cure, and does not say to cure any disease or condition in the body, and merely, as doctor says, allows the communication between the cells and the immune system for our bodies to heal themselves. So, okay, we'll just let you go. We've got about 20 minutes or so of questions. So first in, first serve, just nice and orderly, please. There's a lot of people for Dr. Samuelson. Over to you, Doctor.
Okay, very well. We have about 300 people here, so probably the best way to do this is to, if you can, raise your hand. There is a little, on the side of your name over there, there's a little button that looks like a hand. You can also, I think, put it's on the More menu if you're looking at the participant panel. And if you could, then raise your hand. That'll bring it to the top of my list and make it easier for me to identify those people who have questions. I'll take your question, I guess. Thank you. What's your name?
My name is Bruce Wiseman.
Bruce, good to have you on today.
Thank you. I have two quick questions. The first is, I think, a simple kind of yes/no question. In looking at the a graphic that you provided in a prior webinar, maybe 30 days ago, and then reading some material, it appears that the two kinds of signaling molecules that come out of the mitochondria also come out along with the ATP molecule. They seem to come out at the same time. And I was curious if the consumption of ASEA from an external source, obviously drinking it, would generate more ATP, or is that just something that has to be done via the mitochondria?
There is some upregulation of the mitochondrial activity when they're through the means of this reactive oxygen species. So what you're referring to actually is the mitochondria, when it produces the energy necessary in form of ATP to keep all of your cells running. It also produces the same redox signaling molecules or reactive oxygen species that are in the product. And that's very curious. And then the question is, well, when we drink the product, do we replace then the deficient molecules that are normally produced by the mitochondria when we're younger, and as we age become less and less efficient? So the question is, well, do we actually increase the energy, the ATP? And the answer is, in some of the studies that we had in North Carolina, it seemed that we may have mitochondrial biogenesis, which means that the mitochondria become more prevalent. But that was also confounded by the fact that we used athletes. And athletes, when they exercise in their muscles, also produce more energy. The mitochondria are dividing and producing more energy. So we weren't sure whether it was the ASEA or it was the fact that these athletes were exercising.
I see.
But it is possible that we are producing a little bit more energy. Certainly the regulation is upward. It's not downward. When we drink ASEA, we're not seeing less energy produced.
Understood. One other quick question, and I'll get off. In your prior webinar, as well as watching videos of other of the medical professionals, there is this constant reference to ASEA creating a balance, and I wasn't sure exactly what's balanced when I look at the two kinds of reactive molecules coming out of the mitochondria. It seems that one, by its very existence, brings into existence the antioxidants to deal with it, and then the other one goes into the immune system to help the immune system identify damaged cells. But there is this word balanced, and I go, well, what actually is being balanced? Is it the fact that the newly created antioxidants are now sufficient to take care of free radicals, or what's the benefit there?
Oh, well, that's a really good question, and actually quite a deep question. One of the first ones I asked myself. Because when we had these 16 years, now we have 22 years of studies that show that ASEA has absolutely no adverse effect to the cellular physiology. No adverse effect whatsoever in any of our studies in any of the participants or animals. And the question is, well, why? And the answer is basically that we have the same balance in ASEA as we have inside our fluids inside the cells. They're the same balance. So in that sense, it's perfectly balanced. And it's very difficult, for me to explain this in a way without using too many chemical or confusing terms. But the thing is the body produces these redox signaling molecules from salt and water. Inside of our cells, it reduces and oxidizes salt water in order to produce these elements. And we have a chemical balance in the sense that it can be reversed, meaning that the antioxidants and enzymes then can turn these things back into salt and water. So do you understand what I'm saying?
Yes.
Suppose we just had, for example, hydrogen peroxide, which is one of them, but none of the others. Well, then it would be imbalanced. Suppose we had hypochlorite, it would be imbalanced, and it would cause irritation. It would cause inflammation in of our cells because of the imbalance. But it turns out that ASEA is perfectly balanced because it acts as if it's not even there for healthy, normal cells, and it only acts to help detect, repair, and replace those cells which have become damaged or deficient. So,
do the pardon the interruption, but do the antioxidants created in the cell by the reactive species, do those antioxidants not only, do they deal with free radicals that come from toxins or environmental pollution into the cell?
Yes, they do. But the toxins and the pollutants also stimulate the cell to create reactive oxygen species and more oxidants. That's where the inflammation comes from, is because the cell doesn't know how to deal with these toxins. And so it becomes stressed and creates oxidative stress. ASEA helps those cells then to deal with the oxidative stress more efficiently.
Thank you, Doctor.
You're very welcome.
Doctor, can I ask you a question?
Sure.
I have a quick question. My name is Frank Colabella. How does ASEA specifically increase glutathione in the way that it does? I've known about glutathione. I had my pregnant wife on it 22 years ago because I heard it was a great thing for someone that was pregnant to be on because of how it chelates metals, heavy metals, and pulls them from the body. How does it do that? I find that remarkable.
Yeah, that it is a remarkable thing. There's something called the translocation of NRF2. NRF2 is one of those signaling messengers, by the way, that's created, and it is translocated into the nucleus to push one of these genetic cells, and it pushes the glutathione button. So through the NRF2 pathway, glutathione is produced, is upregulated. They say it's produced more prevalently inside the cell. Well, one of the genes that is expressed, the EGR1, is upregulated. It also helps to produce more NRF2. Some of the early studies done at PNNL in the United States at the credible laboratories have shown also the translocation of NRF2 in cells when ASEA is added. So it's the NRF2 pathway, we believe, that is being activated to create more glutathione as well as more superoxide dismutase in our cells.
Thank you very much.
Thank you.
That's great. Dr. Gary Samuelson, we have got Barbara Kerner who has been patiently waiting for a question. Just a reminder, please keep the questions compliant. Over to you, Barbara.
Thank you. Thanks, Dr. Samuelson. I'm just wondering, some of the people I speak to about ASEA are very keen on scientific validity. I'm just wondering whether this genetic study is going to be published in a peer-reviewed journal at some point.
Oh, that's the million-dollar question. You know, all of the studies we've done so far have not had publications in major journals, but we are sure hoping that they will indeed come out and publish some of these results in a scientific journal. It's not as easy as you may think. It takes many months, if not years, to go through the process of getting published.
Okay, thank you.
Yeah, thank you. Thank you, Barbara. That's great. Dr. Frank, I'll come back to you in a minute. I just got Lewis Baker, who had the hand up as well. Just come line, please.
Yes, Dr. Samuelson. Earlier you did a video, actually I think earlier this year, and you were talking about nanoparticles. You were telling the story of how before you found ASEA, and I was just curious to know how does ASEA compare with nanoparticles? Does it help that science, or is it a greater science with the redox molecules? In other words, is it more of a advantage to put the effort into the redox science in terms of what you were saying those nanoparticles can do? It seems like with redox, it does a lot of that, and it seems like it opens up a door of even more possibilities of application of the product.
I don't believe that there is one type of science that, when we're talking about biology, that has an advantage over another type of science. They're whole categories, you know, so it's a very general question that you're asking. There are areas where nanotechnology can help very specific areas of the body. For example, it can block viruses. And so if you have a certain type of virus in your body, you can use nanotechnology to attack or to attach to these viruses and disable viruses. Well, ASEA does that, but through a different area. It helps the cells in the innate immune system to become more efficient, to detect and destroy the virus through other processes. Now the question is, you know, which one is more important, or which one is best? Well, if you're dealing with the Ebola virus, maybe the immune system and nanoparticles would be helpful, or something that's very. But you know, I don't think that there really is an answer that I could tell you to that question. One thing I can say though is that the redox signaling process is so fundamental to every system and cell and function in the body that it is more general. It's more generally beneficial than a nanoparticle, which would have specific target inside your body in one area or the other. So you know, as far as being more beneficial to the whole body, I would say that the redox signaling wins.
Your chance to ask the question, please.
Okay, so I just had a question, Dr. Samuelson. I was just wondering, I was curious when I saw the results of the study that was done, if we had checked blood levels of those participants after the 8 weeks, would they have had a higher increase in their genetic pathways being turned on? And is it dose dependent? And I understand that it was done with 8 ounces each day, but if we would study a larger dose, would we have seen a larger increase in the genetic pathways being turned on? And I'm just curious about the studies. I would love to see more, and I would love to see, I have so many questions about what could happen if, and so I know this is crazy, and from your standpoint, do you see this being studied further? So.
Oh, well, fantastic! You have the mind. You have the mind of a scientist. Yes, we are.
Yes, I know.
Very, very good. No, I would love to see if there is dose dependence. Unfortunately, that was not done. What was done also, what was interesting is that they did take on week 16 another blood sample to see what happens after they stop taking ASEA for 8 weeks, and found that all levels returned to normal. So that we do know, and no, we don't have dose dependence. My feeling is that absolutely yes, we'd see a dose dependence if we actually did the study, but that's a hypothesis, not a result, right?
Thank
you very much, Anna. That's just lovely. Margot Colasso, you're the last person before we take Dr. Frank's questions again. Margot Colasso. Are you there, Margot?
I hope I can word this. Is it possible to strengthen the existing alveoli that are undamaged in the lungs?
Yes, it is possible. Regeneration is possible in the lungs. In fact, we've seen that ASEA has an extreme effect in what they call the small cell endothelial in cells in the lungs. Those were the ones that we were studied in the national laboratories, and it's very, very effective in those types of cells. My goodness, in the lungs is probably more effective than almost any other part of the body.
Can I say whoop whoop? Thank you very much.
All right.
Thank you very much, Margot. And now to the last question from Dr. Frank, who's been waiting is that right? Do I have you right, Dr. Frank Colabella? You had your hand up before. Are you there? This is your chance.
I was the one that asked the question about glutathione. I kind of rudely interrupted you, and I got my answer. So I'm okay. Thank you.
All right.
Okay, great. There's Ron. Ron is trying to ask a question too.
Okay.
Yes, sir. Thank you very much. Can you hear me?
Yes.
Ron.
Thank you. I have friends that have issues with their myelin sheath being deteriorated. You felt the redox signaling molecules would have promise there for identifying the damage and possibly regenerating the sheaths again so they can function more effectively.
Absolutely. Reduction of oxidative stress will help indeed reduce the amount of damage that's caused collateral to the cells that have been stressed. So often pain, for example, pain sensors and such are triggered by oxidative stress and inflammation. That lets the body know that something is wrong. When you get too high of levels of these oxidative stress, it can damage these myelin sheaths. Reduction of the oxidative stress and repair and replacement of damaged cells is an extremely important process in these cases. Now, the lesions also in the brain are aggravated by oxidative stress. In this case also.
That's very encouraging. Thanks. Oh, one more question. What about Dr. Samuelson? Have you heard anything that might alleviate that issue with people that have the machines on at night and so forth?
Yeah. Yes. There's some reduction of oxidative stress or mental stress helps, and also inflammation. I don't know exactly. We haven't done any studies on it, so I couldn't give you anything really direct on that.
Sure.
Yeah.
Sounds like oxidative stress is the bugaboo in so many of the issues we're all facing.
Yes, indeed. And repair and replacement of damaged tissues is extremely important in the regaining your health. Very well, Maria.
Perhaps I have instructions to ask for 3 more questions, perhaps, or maybe we can read some that are in the chat. Would you like me to do that, Doctor?
Of course, it's your show, Maria.
This has been a wonderful, wonderful webinar. Thank you so much, Dr. Samuelson. It's been very inspiring, and of course, very. It's just awesome to find out that there's so much this technology is ours, and we can actually do something with it. Now, I've got a very long question here from John Flanson. I work as a building geobiologist, specialized in radiation protection and environmental medicine. I wonder, since EMR, electromagnetic radiation, opens up the voltage gate calcium channels, resulting in oxidative stress and 1 and 2 strands DNA damage through the Krebs cycle. Back in 1971, the US Navy already linked EMR to 120 illnesses. I'm trying to fit the puzzle for myself in regards to genes, DNA, and oxidative stress. Thank you.
Oh, wow. I thank you very much, and may you have the greatest success in your career. The person who asked that question. I have inside my laboratory set up a radio transmitter that turns enough to check the voltage-gated calcium channels in tissues to see which frequencies affect the tissues more drastically. And we've seen these type of effects. And yes, the ASEA product is able to reduce oxidative stress inside of tissue that has been stressed through electromagnetic radiation. It's the same as almost as other types of stress. But I think you are right on point in saying that it's the voltage-gated calcium channels that are causing the most problems with us in the environment of the radiation that we have all around us in our environment. I'm trying to identify which frequencies are worst, and then how we can block those frequencies on a physiological level. So we're actually doing work in that in my laboratory. Thank you.
Thank you, Doctor. One more question, and it goes like this. It's from Bernadette. If the hormonal pathways are working, do they influence the reproductive systems?
Do hormones affect reproductive systems? Yes. There you go. Absolutely.
Okay, and the last one, I'm just reading to see if they're compliant. Okay, this is from Clara Tam. For people with overreactive immune system, things that develop allergies, would ASEA redox signaling molecules cause a boost of the immune system, and hence that is causing the allergy?
Yeah, helping to regulate the innate immune system, that is the system that's caused by irritation from outside, as well as pathogens, is something that we know ASEA can and will do. Now, it appears as if we see in the innate immune system regulation that we can normalize immune systems effect. So if we have an overactive immune system, turning the cell phones on, I should say, not the radios, but turning the cell phones on helps the immune system respond better. Whether it be overactive and needs to come back down, or underactive and needs to be activated.
Okay, well, thank you very much. Those were the last three questions. I just would like to say to you, Dr. Gary Samuelson, thank you very much for the opportunity to host you and keep everybody on toes. And look, just the information you provided us and the belief so that we can become and belong, it's been amazing. I thank you so much. Thank you, everybody, for being so supportive. We had an enormous crowd on today, very successful. And on behalf of everybody in RS Samuelson, I would like to say thank you so much to everybody. We're now on the hour. So have a good day.
Could you mention, Maria, that I do have a blog called drgarysamuelson.com, and they can search for more information on my blog.
Absolutely. I don't know whether you actually have all heard that. It's I'll just put it on the chat here in a minute. But Dr. Samuelson has got a fabulous blog. It's very informative, and he keeps it up to date. And it's www.drgarysamuelson.com. And just please feel free to use that information. Also, if you have any other questions, please put them in the chat. Dr. Samuelson will read the chat later on and perhaps get in touch with our upline, as I said before. But right now, thank you so much, and I appreciate your help and your support. This has been a wonderful thing. Thank you, Dr. Gary Samuelson. I appreciate it. Thank you, everybody.
Thank you all.
Thank
you. Thank you, Maria.
Thank you so much.
Thank you.
Thanks very much.
Thank you, and I hope you taste it. I hope we can see it. I hope it will be somewhere that we can really listen.
Thank you, Doctor.
Thank you.
Thanks for all your work.